right? So this is a muscle fiber or a myocyte or a myofiber, so many different terms, same exact structure. It's a cell that is a muscle, right? And so the muscle fibers are surrounded by the endomysium. Again, this connective thing back here. Right? Muscle fibers with their endomysium are surrounded by a paramysium. Right? So lots and lots of muscle cells here, each surrounded by their own connective tissue, bundled into, lo and behold, a fascicle surrounded by paramysium. Just like neurons are bundled together into fascicles and surrounded by perineurium. Right? Nerve for N-E-U-R for neuron or some kind of nervous something. M-Y-S or M-Y-O always means muscle. And finally, lots of fascicles are bundled together into the muscle itself, right? So this, this is one of your quad muscles, actually. And so, the vastus lateralis muscle is bound together by the epimysium, right? And actually, many of us, if not all of us, have seen fascicles before. So if you've ever cooked a piece of meat in the crock pot, or even just had like a pulled pork or a pulled chicken sandwich, all of those little shreds of meat are fascicles, right? So the cooking slowly actually breaks down the epimysium and it leaves this paramysium intact. And so all of those little strings are just bundles of muscle fibers, right? So those are fascicles. Okay. Speaking of which, we have a chicken leg dissection coming up in like a week and a half. Okay, just a reminder. Okay, myofibers, right? Also called myocytes, also called muscle cells, whatever. These are cells just like any other, right? So we've been talking about a normal old human cell, right? A nucleus, mitochondria, RER, etc. Same exact stuff is going on in a skeletal muscle cell. But there are a couple different names, of course, a couple modifications, as you might imagine. First of all, we don't call the plasma membrane of a muscle cell just a plain old plasma membrane. We call it a sarcolemma, and this is another root that you're going to see over and over again. Sarco means muscle cell, right? So myo, mice, and sarco all mean muscle, right? So any terms that you see with those roots has something to do with this cell that we're talking about. Okay, let's see, instead of just one nucleus, there are many nuclei. There are an outrageous number of mitochondria, and there are modified reticula. Okay, so we talked about the RER, the SER. Now we're talking about the sarcoplasmic reticulum, or the SR. All right, we'll look at that here soon. Ultimately, the different structures within myofibers give rise to the function of the cell, right? How to structure match function? Well, myofibers or muscle cells are contractile, right? They can get shorter, right? Never pushing, always pulling. They are excitable. That is, we're going to talk about action potentials again. It's not over yet. In fact, it won't be until the end of the semester. Conductivity, right? So we can respond, these muscle cells can respond to a stimulus and produce this electrical response. Extensibility. Unlike most of our cells, our muscle cells can actually stretch beyond their normal length without tearing, right? As long as everything's working out okay, and we'll get into that at the end of today. And finally, elasticity. Once we stretch those muscle cells out, right, once you do a super stretchy yoga pose, your muscles can return back to their original length, right? So this is very unique among cells, right? Their structures give rise to these very special functions, okay? If we look inside these muscle cells or these myofibers, we will see that they're composed of a lot of different little cylinders, right? These cylinders are called Myofibrils, right? And so there's so much nesting going on with within muscles, right? Here is the biceps brachii. Here's a fascicle. Here's an individual cell. Within the individual muscle cell, we have all of these different cylinders, right? So layer after layer after layer of structure. Okay. The cylinders, again, are called myofibrils. They are pretty much filling the entire cytoplasm of the cell, which again, we have a special name for it inside a muscle cell. It is called sarcoplasm, right? Sarco means muscle cell. So the cytoplasm of a muscle cell is called the sarcoplasm. Okay. These myofibrils are essentially as long as the muscle cell is long, right? So several feet long for certain muscles. And these are what actually make the muscle cell appear striated. Okay. Those cylinders have even more crazy overlapping structures inside of them. Within the myofibril, right, which is part of the myofiber, are overlapping proteins. These proteins in general are called muscle filaments for proteins, so literally muscle filaments. There are several that we need to be familiar with because they give rise to the function of the entire muscle. First of all, we have the thick filaments, right? And they're called thick filaments because they're actually many, many chains of a protein called myosin bundled together. Each individual myosin filament has a little flexible head attached to it that hinges, right? So it hinges and it can attach to, under certain circumstances, to the thin filaments, which are also called actin. And this word should seem familiar. It is one of the cytoskeleton fiber types. Thin filaments don't have huge bundles of protein chains. Instead, they just have two, and a little spiral of actin down here. Okay. Each of these little like balls here on actin has what is called an actin active site or a binding site. Essentially, this little opening can bind reversibly to the myosin heads, right? So when, or under certain circumstances, the active site is wide open. As long as it's wide open, myosin, specifically these little heads here, is going to bind to that active site and start doing its thing. And we'll get into that here in just a second. I say under certain circumstances because there are two more proteins that are involved in this process. First of all, we have something called tropomyosin. And tropomyosin is this little teal strand right here, right? And when your muscle is at rest, right? So your legs right now are pretty relaxed because you're sitting down on the couch. And so right now, tropomyosin is covering over the active sites on actin, preventing the myosin heads from binding to the actin active sites. Okay, the way that we get tropomyosin off of these active sites so that actin and myosin can actually bind together is troponin. Troponin is the fourth and final one of these proteins that's super important in this process. Troponin can literally lift tropomyosin off of the active sites, therefore allowing myosin to bind to actin. Okay, so I know there's lots and lots of terms there. Yeah, lots of terms, but we're going to put them together into a story here in just a moment. All right, so the third and final myofilament, which we're just going to mention and not get into the physiology of, mostly because our understanding of this is still very new. These are elastic filaments, so essentially springs on the end of our myosin that essentially allows the muscle to return back to its original length super quickly. Okay, any questions at this point about that? I know there's a lot. We're gonna start drawing in a moment here. Actually, we're gonna start drawing now before we get too far ahead of ourselves. All right, so get out the paper. Hopefully I can do this with relative ease today. Looks like I lost all the stuff I put up here earlier, but that's fine. Okay, so the process we are talking about now is myofiber stimulation through relaxation. Remember that myofibers can be called myocytes, muscle cell, or even muscle fiber. All of this means muscle cell, right? we'll just say contraction. Okay, so that's the name of this process. Okay, let's draw in what we know already. Okay, so what we have is the plasma membrane of a muscle cell. We know that this is called the sarcolemma. All right, so that's what that line is. We have the sarcoplasm inside, right? So the gooey cytoplasm of a muscle cell. I have drawn for you this kind of, this right here, little indentation. This is called the motor end plate. And this is going to be important because our our motor neuron is going to interact with the muscle cell right here, right? So there's just one motor neuron that is in charge of controlling this particular muscle cell. Right, so this is our motor neuron. Now we know that the motor neurons are exiting out of the spinal cord via that ventral root down through the spinal nerves and ultimately getting to, our leg muscles. Okay, so that's where we are so far. We know also that the sarcoplasm is filled with cylinders. Okay, and these cylinders, I'm not going to draw them all the way across because it's just going to get in the way. But these cylinders are called Myofibrils. Maybe. Okay, yeah, so these are Myofibrils, and in reality, they are filling the entire screen, but I don't want to, get too much distracting stuff drawn in our little diagram here. Okay, also, we have overlapping actin and myosin. Okay, so I will draw the red, right, the thicker red here, as myosin. Okay, we have lots of parallel myosin filaments. Okay, and they are filling all of the myofibrils, which are consequently filling all of the myofibers sarcoplasm. Right, lots and lots of different terms there. Okay, so again, these guys are myosin. And those are the red ones. Okay, the myosin also have these funny little heads, right, on both sides of the fibers. So that the, trying to get so fancy here. So that the overlapping actin, the thin filaments that is, can bind to these little myosin heads. Okay, so actin and myosin are overlapping one another. We can see that some regions have just actin, some regions have just myosin, and some regions, they are capable of interacting with one another, but they're not at rest. Okay, so this blue here, these are actin. Okay, we also know that covering over the actin are tropomyosin filaments. Right, so they kind of weave around actin, covering over the active sites on that actin. Right, and I'm sure you can use your imagination for all of these to be on everything, right? All of the blue, right? So the purple here is tropomyosin, and that covers the active sites at rest. Okay, the only way that it would not cover the active sites is if another protein lifted the tropomyosin up and away from the actin, right? And so those, oh, sorry, too big. Those proteins, the final proteins that we're going to draw in here, are called troponin, right? And I'm just drawing them in as these little orange little guys here. Again, they are a protein that we call troponin. And this removes tropomyosin for contraction to start. And of course, if there are any questions, you just let me know. And I spelled that wrong, but forgive me. Okay. One more thing before we jump back to the PowerPoint here. The actin filaments are connected together to each other, that is, by proteins, kind of zigzaggy here. Okay, so this is again binding the actins together, so they all kind of move as one. Okay, these are called Z discs, right? And Z discs are important because from Z disc to Z disc, right? So from little thing binding actin together to little thing binding actin together, we have a structure called the sarcomere. And the sarcomere is super important because, that's just not gonna work. Because this is the functional contractile unit of the entire muscle. So every time you flex your biceps, your sarcomeres are shortening. So essentially Z disc is being pulled towards Z disc by the action of these little myosin heads binding to actin. Okay, so that's where we're going here. Okay. Any questions before I flip back? Okay. Let's go back to this. Okay, so what we can see here are a myofibril, right? So one of those cylinders inside the cell. We can see overlapping filaments here. And if we zoom in, we can see actin is in blue. We can see the thick filaments myosin with all their funny little heads is in red, just like our diagram. Here's titin, which is that spring to help the muscle recoil, and Z discs. Right, so these are the Z discs marking the ends of what is called the sarcomere. Okay, now when we look under the microscope, which we're not going to do for this section of the class, but when we look under the microscope, we can see that there are bands of dark and light and darker yet. This is pretty much showing us essentially where we have regions of overlap versus just actin versus just myosin. And just like with anything else in A&P, we have names for all of these different regions. These regions are ultimately what give rise to the striations of the muscle. Okay, so the A band, for example, is the entire length of the myosin. Right? So from the end of myosin to the end of the myosin, this entire thing is the A band. Within the A band, we have the H zone. The H zone only has myosin, right? No actin, right? The A band has actin in some places and just myosin in other places. The H zone only has myosin. Within the H zone, we have the M line, and so this is the middle line, the middle of the sarcomere. Also within the A band, we have the zone of overlap, and that is a really important one to know because this is the only area that is available for myosin to bind to actin and therefore give rise to shortening and tension or strength in those muscles. Okay? So when we stimulate the muscle, which we're getting to soon here, these little myosin heads are going to bind to actin only if actin is right there. Right, so only in this area of overlap. Next, we have the I band. The I band only contains actin, right? So where the myosin stops to where the next myosin starts, this here is the I band. And of course, within the I band is the Z disc. Okay, so Z disc here and Z disc here, both within their respective I bands. And of course, from Z disc to Z disc is the sarcomere. Right? If you remember any of these things, sarcomere and zone of overlap are by far the most important. Okay? We are shortening sarcomeres. And in fact, I'm doing a lot of arm waving here, but essentially I will keep saying that this is like a sarcomere, right? So my thumbs here are the Z discs. There's a zone of overlap in my fingers here. And so when cross bridges form, we are going to have these filaments sliding along one another and essentially moving your thumbs closer together, right? So lengthened sarcomere, shortening sarcomere. So when you have a lot of these doing this at the same time, that's going to shorten your entire muscle. Okay. Again, the functional contractile unit of the muscle is a sarcomere, right? So even though our entire muscle is contracting, this action is occurring at these super tiny structures called the sarcomeres. Okay, and so again, the big picture, where we are going with this is that when our muscles are stimulated to contract, so when you think, hey, move my arm, your arm moves. What's occurring in that very brief period of time between, I want to move and I move, is we stimulate the muscle and essentially, those myofilaments, actin and myosin, start sliding along one another, shortening all of your sarcomeres simultaneously. And so what this is showing us over here is at first there's a relatively small zone of overlap. Okay, these little myosin heads are going to grab onto actin and they're going to start pulling the actin towards the M line. Okay, and so after doing this for a brief moment, you're going to see that the Z discs are closer to the M line, of course, closer to each other. The contracted sarcomere is now shorter than it used to be. Okay. I don't think it'll let you hear it if I play it from that. So let me show you the video just so that you can visualize what we're talking about here. All right.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so that's pretty much like the whole process that we're going to be walking through here, just as a little preview so you can visualize where we're going. Okay.
Muscle contraction
Oh no!
is at the basis of all skeletal movements. Skeletal
I have
muscles
no
are
idea where that's
composed of
coming
muscle
from.
fibers which
Okay, sorry about that, guys. Assuming you can see this again. Okay, so in your book, you have this image. Again, it is just giving you a little bit of perspective here on, you know, the different nested layers of muscle, right? The whole muscle, the fascicles, the muscle cells. Within the muscle cells, we have myofibrils. Within the myofibrils, we have overlapping myofilaments. And that is the level that we're talking about. These sliding filaments, right? So myosin actually binding to actin and pulling it over, detaching, binding again, pulling it over, and so on and so forth, bringing those Z discs closer together. Okay, so as you saw in the video, there were a couple other things that were important for the actual functionality of the muscle cell and its sarcomeres. Okay, so in addition to having these myofibrils filled with myofilaments, which by the way on our model here, the cylinders are myofibrils, these little lines here are myofilaments. We also have these deep invaginations of the sarcolemma, right? So the outer membrane around the cell that actually pass horizontally for the most part through our muscle cells. Right, so essentially the same phospholipid bilayer that is on the outside of the cell dips down inside the cell, essentially creating a little pathway or a little environment that is the same as the outside of the cell's environment. That is, outside of these muscle cells, there is high sodium, low potassium. These T-tubules have the same exact concentrations, high sodium, low potassium. So it's essentially taking the extracellular environment inside the cell, but not inside the cell per se, right? It's still isolated inside. Okay, and so on this diagram, we can see the little T-tubules right here. On our model, the blue are the T-tubules. Okay. Also, we can see that the T-tubules are surrounded by this funny little mesh thing going on. This mesh is actually a modified reticulum, right? Modified smooth endoplasmic reticulum, in fact. This is called the SR or Sarcoplasm Reticulum. It is wrapping around every single myofibril, and it is going to be interacting in a particular way with those T-tubules. In fact, if we look closely, we can see that the SR, which is this big like spider web looking structure, gets pretty fat as it gets close to the T-tubule, and it gets fat on both sides. These fatter regions of the SR are called the terminal cisternae. And if you take off the E, terminal cisterna is singular. Okay, together, both terminal cisternae on both sides of the T-tubule and the T-tubule itself make up what is called the triad. Right? So terminal cisterna, T-tubule, terminal cisterna, collectively called the triad. Okay. And so what the heck is the point of this? Well, at rest, the membrane of the sarcoplasmic reticulum, right, so this blue stuff right here, is actively pumping calcium out of the sarcoplasm, right, so out of just like floating around the muscle cell and into the SR. Right, so what these little arrows are representing is active pumps, right, pulling calcium inside the SR. Okay, and now it's separated by a membrane, and so inside the SR is unbelievably high calcium levels. In fact, it can be 40,000 times greater calcium levels inside this SR than it is outside. Okay, so huge concentration gradient here, right? We are setting ourselves up for a really rapid diffusion of calcium out of the SR. To be continued. Okay. Also, we have voltage-gated calcium channels, right? So we have pumps pulling it in. We also have voltage-gated calcium channels that are embedded in two membranes. First of all, they are embedded in the sarcoplasmic reticulum membrane, right? So in this place that is chock full of calcium, but there's a little piece of these voltage-gated calcium channels that is actually embedded within the T-tubule membrane as well. And so essentially any significant change in charge, that is depolarization, that comes down the T-tubules, right? So in other words, when an action potential comes down the T-tubules, this is going to change the shape of the voltage-gated calcium channels and let calcium flow with its concentration gradient, which, as we know, is out of the SR and into the muscle cell. And remember that, just right underneath the SR are these myofibrils, which are which contain actin and myosin. Actin is covered with tropomyosin, and unless there's calcium, tropomyosin is going to stay over those active sites, right? So we're getting there. Okay, so again, at rest, we are building a huge concentration gradient of calcium inside the SR. We can already see that calcium is going to be released from the SR super fast when there is a change in charge. So when there is a depolarization, the depolarization of an action potential. We also have another set of concentration gradients that are being generated at rest. Just like in neurons, we have a resting membrane potential in our muscle cells. This resting membrane potential is actually even more negative than it is in neurons. It is -85. And that's really important because we don't want our muscles just contracting at will. We want to make it really hard for them to contract so that when they do contract, it's at the right time. Okay. And again, same kind of situation as neurons. So I hope that neuron process is still in the back of your mind. Okay. In order to actually get a muscle contraction, we have lots of different things that have to happen. First of all, we have to stimulate the muscle fiber. We have to tell the muscle that it's time to contract. Okay? There are two steps within that, and both of those should be pretty familiar to you. Phase 2 is what is called excitation contraction coupling. So essentially, the action potential, this electrical stimulus, has to be utilized in some way to start contraction, right? So this change in charge, it has to be converted into some kind of mechanical movement, right? So that's phase 2. All of that stuff happens and still we don't have muscle contraction. Muscle contraction doesn't happen until phase 3, which is called the cross-bridge cycle, which you guys just saw in that video a minute ago. Okay. So we're going to break this down. I'm going to walk you through the first 2 steps right here, and then we'll go back to our diagram and add in what we now know. Okay. So phase 1, just like talking to the muscle cell, the very first thing that has to happen is our friend action potentials in the motor neuron has to lead to the release of acetylcholine, a neurotransmitter, into the synapse between the motor neuron and the muscle cell. Okay, so what we can see in these images is a motor neuron. Here is a myelin sheath. It's in the peripheral nervous system, so this is a Schwann cell. Okay, the change in charge reaches the axon terminals, and if we look inside those axon terminals, we know that there will be calcium entering the axon terminal, leading to exocytosis of acetylcholine from the synaptic vesicles into the synapse. Right, and as we drew on our little diagram, there's a little depression in the membrane of the muscle cell. The membrane itself is called the motor end plate. Okay, so here's the motor end plate. The space in between is called the synapse or the synaptic cleft. And one more term, this entire area here, right, the axon terminal and the motor end plate, this whole thing is called the neuromuscular junction, right? So the junction between the muscle and the neuron, sometimes abbreviated NMJ. Okay. Next, step number 2 here, just like with a postsynaptic neuron, we also have acetylcholine binding to these specialized receptors on the motor end plate. Sodium channels open, sodium comes into the cell, there's an influx of sodium, and that is going to depolarize the local area of the cell. So in other words, we just created a graded potential in, or right next to the motor end plate. Okay, so the same exact thing is happening here as we saw in the neurons. Okay, so let's hope that all my drawings are still here. Hooray. Okay, so before we add in these steps, let's add the extra structures that we talked about. Okay, we know that there are T-tubules that essentially are invaginations of the sarcolemma, right? So they are going to bring the extracellular environment inside. Okay, and so this is called the T-tubule. Okay. On either side of the T-tubule, and in fact, covering over the myofibrils and myofilaments and all the stuff that we've drawn already, we have Sarcoplasm Reticulum, right? It is covering over all of this crazy stuff, okay? And just like with the myofibrils, I'm not going to draw it everywhere, right? That would just be way, way too much stuff, okay? But this is the Sarcoplasm Reticulum, okay? And I'm not going to draw in all the holes and anything like that. There are some membrane proteins that are important. Let's see. So we have receptors on the motor end plate. Okay, so these are acetylcholine receptors, and we know from studying neurons that they are associated with sodium channels. Right, so these are our acetylcholine receptors. Okay, and of course they have specialized little areas that specifically and tightly bind to acetylcholine. Okay. We also have the same things that we've seen before on neurons, voltage-gated sodium channels. Although this time the voltage-gated sodium channels are not only along the surface of the sarcolemma, they also are down the T-tubules. Okay, so the T-tubule is essentially the same exact thing as the outside membrane, the sarcolemma. Okay. So again, these are voltage-gated sodium channels. We also have voltage-gated potassium channels, right? Depolarization and repolarization works literally the same way in muscles as it does in neurons. I'm not going to draw that in because there's already way too much going on. Okay, so voltage-gated sodium channels for depolarization, just keep in the back of your mind there are also potassium channels as well. But what I will draw in is these other types of voltage-gated channels, which have a piece of this protein within the SR membrane, and they also have a piece, all right, within the SR membrane. Okay, so essentially what this silly little diagram is showing you is that any kind of voltage changes over the T-tubule membrane is going to change the shape of the protein, which is going to open up the SR, right? So that's really all that that is drawing. And of course, these are all over the place, okay, down the T-tubules. Okay, hopefully this is logical, and if not, speak up. Right, so the green ones here are voltage-gated calcium, and the 2+ is just, it's missing 2 electrons, right? Channels. Okay. And right, that's what these are right here. Okay. Also, while I'm here, this is the T-tubule. Let's see, anything else that I missed? I think that's pretty much, not quite yet. These are the synaptic vesicles, right? Remember, they are chock full of acetylcholine. We'll say is this color. All right, so acetylcholine are all of these little guys in here. Okay. Okay, one more, sorry. The neuromuscular junction, NMJ. It's not going to let me move that, but that's okay. This entire section here, all right, so the neuron all the way through the motor end plate, that entire thing is the neuromuscular junction, the NMJ. Okay, I think we are finally to the point here where we can start drawing in steps. Okay, so step number 1, I'm not going to spell it out in a lot of detail because you have all of these steps already printed for you in the PowerPoint, but step number 1 is essentially an action potential in the motor neuron. So we know that an action potential shoots all the way down the axon, right, from the spinal cord, all the way down to the axon terminal. This leads to an influx of calcium and ultimately an exocytosis of acetylcholine, right? So all of that is number one. The action potential in the motor neuron releases acetylcholine into the synaptic cleft, right? Number two is the acetylcholine is going to diffuse across that synapse and it's going to bind to the receptors on the motor end plates. Okay, and again, there are more thorough descriptions within the PowerPoint. For now, we're just going to draw the acetylcholine in the synapse, and it's going to bind to these receptors. And as we know, when ligands bind to receptors, this is going to lead to some kind of pre-programmed response. In this particular case, it is going to lead to an influx of sodium into the local environment of the cell. All right, so lots and lots of sodium. In other words, a graded potential, right? So we have just depolarized this particular area of the cell. Okay, so it is now more positive here than everywhere else, which is negative 85. That change in charge, if it is strong enough, is going to lead to these voltage-gated sodium channels opening up, right? Therefore stimulating an action potential, right? Opening these up, and all the way down the T-tubule. Okay. All right, so that is steps 1 and 2, or phase 1, where we are pretty much, where we are pretty much just stimulating the cell. Okay. I have embedded all sorts of videos in here because I feel like this is kind of a rough process to just visualize on your own. We are not going to watch all these things, but I highly recommend it, either via this PowerPoint, going to the YouTube link, or it's actually on Mastering A&P as well. Okay, so what we just looked at was an action potential down the motor neuron, influx of calcium, exocytosis of neurotransmitters, acetylcholine binds to receptors on the muscle, influx of sodium causes a graded potential. Now, if that graded potential is strong enough, right, so that was all the stuff over here, what's going to happen is that those voltage-gated sodium channels are going to fling open their gates and allow sodium to flood into the cell. Okay, that will depolarize the next area of the cell, opening up the next sodium channel, depolarization, opening up sodium channel, depolarization, and so on and so forth. And so this action potential is virtually the same as what we saw in neurons, with a few exceptions. First exception is that the action potential is actually going to travel in both directions. Remember that the action potential in neurons only went from axon hillock to axon terminal. Here, the action potential goes every which way across the surface of the muscle cell, including down the T-tubule. And so that is step number 3 in action potential down the T-tubules. Step number 4 is where things start to get a little bit different, right? So in step number 4, the action potential changes the voltage within the T-tubule, which consequently changes the shape of these voltage-gated calcium channels in such a way that they open up and allow calcium to flood out of the SR into the sarcoplasm, into the regular old muscle. And if you look closely here, kind of faded, but essentially, the myofilaments, right, actinomyosin, are right underneath all of this stuff that's happening, right? So dumping out calcium from the SR is literally dumping it right on the things that actually need them. so we'll add that in to our diagram. All right, step number 3. I don't know why the lights are freaking out, but maybe because it's storming like crazy outside. Awesome. So step number 3, the action potential goes everywhere, including into the T-tubules, and that is largely the same. Okay. Wonder how my little baby chickens are doing right now. They are 3 weeks old and outside, yay. Anyway, so action potentials are going to travel along the surface of the cell in all directions. All right, so they're going to go this way as well. Oh, what am I doing? Drawing with the mouse, oh my gosh. Action potentials are also going to go down the T-tubule because of these voltage-gated sodium channels. All right, so that is number 3. Number 4 is voltage-gated calcium channels open up. And this allows calcium into, or I guess we'll say onto, the myofilaments. Okay, so it's actually going over here, right? So dumping out of the SR here, and ultimately it's bathing over all of these myofilaments here. See, so we'll do calcium as brown. All right, so calcium is going to flood out of the SR, which we know is over here. So now there's calcium floating all over the place. Okay. Okay, next. Calcium is going to bind to troponin. Remember, troponin is the type of protein that is responsible for controlling whether or not those active sites on actin are actually open or not. Okay, let's see, this here is troponin. And so whenever there is calcium in the sarcoplasm, calcium binds to troponin, and troponin, as we can see here, is literally going to roll the tropomyosin out of the way. And lo and behold, these active sites were right underneath that tropomyosin, so now they're wide open. Anytime these active sites are not covered by tropomyosin, those little myosin heads are raring to go. They are going to bind to those active sites and start the cross-bridge cycle, as we saw in that video. Okay, so let's go back and see if we can draw this in. All right, so this is step number 5. Now we'll go to this side over here. Calcium binds to troponin, which removes tropomyosin. And that is going to allow the cross bridges to form. And keep in mind that the cross bridges are just the actin and myosin actually being bound together. All right, so myosin binds to the active sites on actin. Okay, so all that's going to happen over here. All of this, right, is at rest. Oh gosh, sorry. So all of that is at rest. Now that there is calcium here, essentially the tropomyosin is going to be rolled out of the way. And now the myosin heads are going to bind. Right, they're going to form cross bridges with the actin. Right, so before all of these, all the myosins above, they're not actually touching the actin. Now they are touching the actin. Cross bridges have been formed. Okay, yeah. So once the cross bridges are formed, then you're going to get into step number 6, and there's no way I'll be able to draw the cross bridge cycle on this diagram, but you could probably draw it by hand if you were so inclined. But let's just take a look at it on the PowerPoint for now. So the cross bridge cycle, as the video showed you, takes place in 4 steps. Once the active sites are open, so tropomyosin has now been rolled out of the way by troponin, that is going to allow the myosin head to bind. Now, in order for the myosin head to bind, it actually had to be kind of prepared for that to begin with. And so the preparatory step for, getting myosin heads ready to bind to actin is this step right here. So the myosin head is cocked, right? It's ready to go, ready to bind to actin. When ATP breaks into ADP and phosphate, right? Remember that ATP is the high energy form. Remember that the high energy bond is between the 2nd and 3rd phosphate here. So as soon as ATP loses this phosphate, that little burst of energy is going to change the shape of myosin so that it actually is ready to bind to actin. Okay, so before the muscle cell is stimulated, ATP breaks, right? It's hydrolyzed, it's ready to go, it's in the cocked position, and then it binds to actin as soon as tropomyosin moves or is moved out of the way. Okay, so this is step number 1. Myosin head is cocked and ready to go. Once the active sites are open, the myosin binds to the active sites. Next is the most important part for the contraction's sake. Here we go. Here's the actin, here's the myosin head. It binds to the actin, and in order to get contraction, you actually have to rotate or pivot this myosin head. And so that's what's going on down here. The myosin is moving. It is the power stroke pushing this actin along, which of course, with all of these myosin heads doing this all at the same time, it is going to pull the actin closer together. Okay? So that's the power stroke. Now, in order to detach myosin from the actin, we need another ATP. Okay, and so actually the active part of this entire process is breaking the cross bridges, so breaking the bond between actin and myosin. Right, so here we can see ATP coming in. It breaks the bond between the active site and actin. That same ATP is hydrolyzed into ADP and phosphate. Right, so it's ready to go again. If there's still calcium in the sarcoplasm, this cycle is going to complete itself again, right? The high energy or cocked position of the myosin head is going to then bind to the actin active site. It is going to pull the actin over. ATP comes along and it is going to break that bond. ATP breaks into ADP and phosphate, right? Recocking the head and so on and so forth, around and around and around. Again, as long as there is calcium in the sarcoplasm. Okay. Again, I highly recommend watching these videos, but I know that we are running short on time. I don't want to keep you here all day. And I recognize that it's already been 2 hours, and if you have other things that you need to do, I totally understand if you have to run at this point. There's a few more slides I just want to wrap up. I will post this video, but I'm giving you an out if you need to run because you have other things going on with your lives. I recognize that. It's all good. Thank you very much for coming by today. Please be careful out there for any number of assorted reasons if you are heading out into the world. Okay. Again, I will post this on YouTube and give you guys the link as soon as I possibly can. Okay, just another way of looking at this cross bridge cycle, right? So before the muscle cell is even stimulated, we get this cocking of the myosin head. As soon as tropomyosin is out of the way, ATP or myosin head binds to the active sites. Here we have the power stroke, the actual movement, the shortening of the sarcomere. ATP comes along and breaks that cross bridge. That same ATP is hydrolyzed to recock the myosin head, and again and again and again. Okay, so the actual steps that are physically generating tension, that are physically moving your muscles, is step 6, the cross bridge cycle, right? So that's the only thing that's actually generating tension in the muscle. That's the only thing that's actually doing the shortening. Right, so, oh, sorry. No, come back. All right, so here's a muscle at length, sarcomere, Z-disc to Z-disc. Here are the little myosin heads. As soon as the cross bridges are allowed to form, they start pulling in the actin, so the actin is getting closer and closer to itself, and so too are the Z-discs. Right, again, this is happening over and over and over and over again. Okay, another way of looking at this, which is actually what you guys will be looking at more tomorrow in lecture as well as in the PhysioX exercise, is this. This is called a myogram. And in the PhysioX, you're going to be looking at what's called an oscilloscope, essentially reading the tension that is generated by a muscle cell or an entire muscle given different conditions. So that's your experiment tomorrow. But this particular myogram, so myo for muscle, gram for like measurement, is showing you how much tension, right, how much the muscle has shortened with a single stimulus, a single action potential. Right, so this is time, this is tension or how short it has gotten. And here is the stimulus. So here is when the motor neuron releases acetylcholine. Okay, note that there is a brief period of time where there's no tension whatsoever that is generated by this particular muscle cell. And so what's happening here is all of those crazy steps that we literally just talked about, right? So steps 1 through 6, we don't actually start shortening the muscle until the cross bridge cycle is allowed to begin, right? So acetylcholine binding to receptors, graded potential, action potential goes down, the T-tubules opens up calcium channels, calcium floods out of the sarcoplasm reticulum, binds to troponin, troponin removes tropomyosin, myosin binds to actin, and finally we have cross bridges and can start to generate tension, right? And so you see just a little bit at first, and so here all the sarcomeres are getting shorter and shorter and shorter, and as long as there is no other stimulus at that point, we're going to see a relaxation period as well. So this is when calcium is being pumped back out of the sarcoplasm, back into the SR, and therefore tropomyosin rolls itself back over the active site. Okay, this very brief, imperceptibly brief, but nevertheless exists, period between the stimulus, the neuron releasing acetylcholine, and the actual generation of tension is called the latent period. Okay. All right. Again, not going to watch this, but I highly encourage you to do so. These are literally embedded within the PowerPoint. Muscle relaxation. Right, so as long as you don't keep stimulating the muscle, which you're going to do tomorrow, but as long as you stimulate it and that's it, the muscle cell has to return back to its original configuration. And so just like we saw with neurons, we have our friend acetylcholinesterase, which comes along, breaks apart acetylcholine, recycles as much as physically possible, which essentially is going to keep it so your muscle doesn't keep contracting over and over and over again. Okay, we'll come back to that in a moment. Also, we have calcium being pumped back out of the sarcoplasm into the SR, right? So more and more calcium is removed. So when you look back here, you'll see that the relaxation period is pretty long, relatively, again, imperceptibly so, but relatively long, because in order to get this relaxation, we need to keep pumping calcium back out of the environment so that, troponin can allow tropomyosin to roll back over those active sites, right? So that's an active process. It takes a little bit of time. And finally, the way the role titin plays in this entire process, right, one of those 3 different types of myofilaments, is that it essentially acts like a spring, and it helps you to return your sarcomeres to the right length. Okay. Final things I want to point out today are a couple clinical conditions that affect muscles. And at this point, we should understand how these work, given what we learned yesterday and today. Botulism is a toxin from a bacteria. You can get this unintentionally by, for example, eating super old canned goods. They love those things. And other types of food contamination. Alternatively, you can get Botox injection, right? Botulism toxin injected for various reasons, sometimes therapeutic, sometimes cosmetic. But essentially this paralyzes muscles, right? And so this this toxin, right, it blocks acetylcholine release at your neuromuscular junction. So without acetylcholine, you don't get the stimulation of those muscles, you don't get action potentials, contraction, cross-bridge cycles, etc. And so this is flaccid paralysis. And so essentially you can think all you want, like contract your muscles, contract your muscles. That message is never going to get to the muscle, right? Even if your neurons are firing, your muscles aren't actually going to receive this message. And so this is important, or this toxin is used in very specific amounts for cosmetic and therapeutic purposes to essentially make it so your muscles don't contract, right? So if your facial muscles aren't contracting as much, then maybe you don't see the wrinkles as much. Or maybe you injured a muscle and you need to give it time to heal. And so you might need Botox to kind of calm those muscles down so they don't keep hurting themselves. And a lot of other reasons. Polio. Polio is a virus, for the most part, was eradicated due to vaccines. Now it's coming back because of opposition to vaccinations. But regardless of that, polio is a virus that attacks motor neurons of the brain and the spinal cord. And so this results in permanent paralysis, right? So if you don't have the motor neurons to tell your muscles to move, they're never going to get the message, they're never going to move. And so again, this is flaccid paralysis. Flaccid as in their muscles aren't going to contract, they're never going to move, as opposed to spastic paralysis, which is where your muscles contract and they don't relax, right? Either way, you're not moving according to your own will. You're either locked up or you can't move at all, or you're completely relaxed. Okay, tetanus. This is another bacterial toxin. It is different than the tetanus that you are going to learn about tomorrow, which is a natural process and we all experience it every day. But tetanus, the toxin from a bacteria, is usually acquired by interacting with kind of dirty substances that get kind of deep into your tissue, so like deep puncture wounds. These bacteria live in anaerobic environments, and so they really like to live deeper within your tissues. Essentially, tetanus is a spastic paralysis, and essentially this results from an inhibition of your inhibition. So essentially, most of your central nervous system would fire uncontrollably, right? Would just spontaneously depolarize and generate action potentials and therefore lead to lots and lots of muscle contractions, except for the fact that we have wonderful neurons called GABA neurons. They essentially suppress a lot of this, or all of this unnecessary, undesirable movement. And so a lot of our, following social norms and whatnot is because we inhibit the stuff that's just not appropriate. And it's, our brain decides what's appropriate, not. And so if this toxin starts to break down those inhibitory neurons, your brain, your central nervous system is going to start firing, start sending a lot more motor stimuli out your motor neurons, and therefore your muscles are going to be triggered to start contracting uncontrollably. And so again, this is spastic paralysis. Your muscles are contracting and they just can't relax. And so this is another one that was more or less eradicated in humans. Not our pets, because our pets generally don't get vaccinated for tetanus, but either way, it's starting to come back once again. It is fatal because, again, your diaphragm is a skeletal muscle, and we need to be able to contract and relax their diaphragm. in order to breathe. Okay. All right, next, myasthenia gravis. This is a loss of acetylcholine receptors at the neuromuscular junctions. So essentially, your motor neurons can be releasing as much acetylcholine as they physically can, but if there aren't receptors on your motor end plates, your muscles aren't going to receive that message. And so you get weaker and weaker because that signal gets weaker and weaker. All right, next, this isn't a clinical condition, but it's kind of an interesting phenomenon that we should recognize at this point. Rigor mortis, right? So generalized muscle contraction shortly after death, so within a few hours. And so if you are a police officer, an investigator, whatever, you can actually figure out how long post-mortem it's been for a victim, etc., given how much rigor mortis they're demonstrating. And so why do people get stiff after they pass away? And so it has everything to do with the cross-bridge cycle and what the actual active part of the cross-bridge cycle is. And so remember we talked about how ATP has to kind of come into the area and bind to this myosin head, and it is the binding of ATP to the myosin head that actually breaks the bond between actin and myosin, right? Cross bridges actually are maintained in the absence of ATP. And so what the heck, how does this connect? Well, when you die, not all of your tissues stop working at the same time. Some of your cells are going to keep on doing what they do until they run out of oxygen, until they run out of resources. And of course, that will happen fairly quickly, depending on the tissue, just because when your heart stops, your circulation and delivery of oxygen stops as well. And so once the muscle cells stop receiving oxygen, they can burn through a little bit of ATP that is stored within them. But once that ATP is gone, essentially, there's no more ATP to break these cross bridges. And as a result, the sarcomeres are kept in their contracted state, and therefore you get stiff, okay? And so ultimately, rigor mortis doesn't last forever, of course. The reason why your muscles ultimately are able to relax and you're able to be a little bit more pliable is because the proteins themselves actually start breaking down. So these myosin heads start to degrade in the absence of oxygen. And so within several hours, the rigor mortis will disappear. Right? The things you may have never thought about before. The last condition I want to show you guys, and it's a YouTube video, so pardon me while I switch gears a little bit here. But this is muscular dystrophy. Let me show you a short clip of this video, and then we'll go back for our final slide of the day.
As children, we have difficulty walking. As teenagers, most require wheelchairs. Their average lifespan is less than 30 years old. What devastating disease do these people have? Join me in this episode of Medicurio, where we will discuss one of the most debilitating muscular diseases, Duchenne muscular dystrophy. All movement is controlled by contracting various muscles in our body. If we zoom into muscle, we see that it is made up of many long tubular cells called myocytes that can contract to cause flexing. An important structural protein called dystrophin is located near the membrane of myocytes. Dystrophin acts like a chain that links the skeleton of the myocyte, made of actin, to the extracellular matrix, which is a mesh-like structure outside the myocyte. This linkage prevents membrane damage when the muscle contracts. Dystrophin has three important areas: the actin binding end, which attaches to the cellular skeleton, the central rod, and the dystroglycan binding end, which attaches to the dystroglycan complex within the membrane that is anchored to the extracellular matrix. In Duchenne muscular dystrophy, A genetic mutation causes dystrophin to be extremely short, often lacking the dystroglycan binding end, making it dysfunctional. Because of this, every time the muscle contracts, small rips appear in the membrane. These small rips allow diffusion of various molecules into and out of the myocyte. The most important substance involved in damaging muscle is calcium. Calcium ions, found plentifully outside of the myocyte, flow in through these small rips and activate calcium-dependent cellular enzymes that break down proteins, called proteases. Normally, by carefully regulating cellular calcium levels, these proteases only break down old and damaged proteins. However, in DMD, extremely high calcium levels activate too many of these proteases, which begin to break down important functional proteins as well. This kills the myocyte. Another important molecule that diffuses through the rips is creatine kinase, which leaks out of the cell and eventually into the blood. This elevated level of creatine kinase in blood is often used to diagnose DMD. Creatine kinase is an enzyme that stores energy for myocytes to use during contraction. With less creatine kinase, less energy storage occurs, which also weakens muscles. Muscle repair and regeneration can occur at younger ages. As patients get older, though, muscles no longer regenerate fast enough to keep up with the constant death of myocytes. Instead, fat and scar tissue begin to fill in the gaps. Since fat and scar tissue are unable to contract, muscles get weaker over time. This weakening leads to a distinct pattern of symptoms, such as Gower's sign, where a child must use his or her arms to stand up because the leg muscles are too weak. As well, other physical symptoms appear, such as a curved posture to account for weaker chest and leg muscles, and calves that are swollen due to buildup of fat and scar tissue. Since the heart and diaphragm are also muscles, they gradually weaken over time as well. Eventually, they stop working, leading to death often before age 30. Currently, there is no cure for DMD, but there are many ways to control its symptoms to prolong lifespan. Physical therapy, steroids, and surgery can all help slow down muscle weakening. What is most interesting is current research being done to cure DMD by directly manipulating the dysfunctional dystrophin gene. In order to understand how these techniques work, we must first understand the characteristics of the dystrophin gene.
Okay, and of course,
the dystrophin gene is located on the X.
So we won't get into that. Although we will talk about how the dystrophin gene is located on the X chromosome. So it's located on the X chromosome, which means that DMD is by far much more common in boys than it is girls, because males only receive one X chromosome and females receive two X chromosomes. And so if this recessive disorder, or if a female gets a recessive allele for this disorder, then she still has a dominant allele to make this dystrophin gene the way it's supposed to be. But males, of course, only get one, and therefore, if they get this recessive allele, they have to express it. And so I just wanted to point that out because, well, it kind of ties together a couple things. In the beginning of this, this half of the lecture, I told you that muscle cells are really stretchy and they can recoil, and that's really unique about muscle cells. In patients with this genetic mutation, just one tiny difference, like 1 or 2 nucleotide difference, leads to a different amino acid, which leads to a shorter dystrophin protein, which leads to the inability of these muscle cells to both stretch and then recoil again, and it is a fatal disorder. And so this is just one of many examples of how even a tiny genetic mutation can lead to profoundly awful consequences here. And of course, this video goes on to talk about some possible treatments. The gene therapy that he showed you at the end is actually like putting the right dystrophin gene into a little empty virus, and then the virus actually injects the gene into the right cells. So obviously, lots and lots of research has to go into that before it's actually used on patients. The link at the bottom shows some pretty exciting developments in studying muscular dystrophy in dogs. They can actually walk and run and play and everything after getting this treatment. So hopefully we can expand that process out to applying to humans. Also, the FDA and patients, parents, and all sorts of folks have been going back and forth over another treatment which may or may not actually be helping, and it is outrageously expensive now that it's not within the R&D phase. And so it's an interesting story, and I can post that article on Blackboard if you guys are interested. But this is a profound disease just based on one tiny little mutation in a protein. Okay.